Assalamualaikum and hi. Today we will learn chapter 2, cell structures and functions. So there are 4 subtopics in this chapter. First is 2.1, prokaryotic and eukaryotic cells. 2.2, structure and function, cell membrane and organelles. 2.3, cells are grouped into tissues. And 2.4, cell transport. So for today, we will learn 2.1 and 2.2 only. Okay, learning outcomes for 2.1. A. State the 3 principles of cell theory. B. Explain the structures of prokaryotic and eukaryotic cells. So these 2 learning outcomes has been covered during lecture. So today we will focus on C. To illustrate and compare the structures of prokaryotic and eukaryotic cells, which are plant and animal cells. Okay, this is a diagram for prokaryotic cells and eukaryotic cells. So for prokaryotic cells, or also known as bacteria, so this is the typical structures of the bacteria. As you can see, it has a structure that we call as flagellum, pilus, capsule, cell wall, plasma membrane, fimbriae, cytoplasm, ribosome, nucleoid, inclusion, and plasmid. We will explain about all these organelles and structure after this. Okay, for eukaryotic cells, this one is the structure of the plant cells. So this is all the organelle and structure that can be found in plant cells, and this one is the structure of animal cell. Okay, prokaryotic cells. What is the definition or the meaning of prokaryotic cells? So it is actually cells that have primitive nucleus. So primitive here means that early or first. Okay, so or nuclear material that is not enclosed by nuclear membrane. So karyo is something that have to do with nucleus. So what we can say that prokaryotic cell is a cell that have primitive or early nucleus, or the nuclear material is not enclosed by nuclear membrane. So of course, if it is no nucleus, so the nuclear material, for example DNA, is not enclosed by nuclear membrane. So this is the diagram of the bacteria. Next is eukaryotic cells. For eukaryotic cells, it is defined as cells that have true nucleus or nuclear material that is enclosed by a nuclear membrane. So, differ from eukaryotic cell have true nucleus, which means when it have nucleus, the nuclear material or the DNA is enclosed by nuclear membrane. U from the eukaryotic cell means true, and karyo something to do with nucleus. Of course, it has true nucleus. So for example of the eukaryotic cells like animal cells and plant cell. Okay, so this is the structure, the important structure found in prokaryotic cells. Okay, so we zoom up to the structure of the prokaryotic cells first. Okay, so look at the first one. Okay, capsule. So capsule, this structure, it is like a greenish blue color. So it is the outermost layers of the some bacteria. So capsule is like a structure that is function to protect the bacteria from the host immune system. Okay, for the second layer after the capsule, the silver color here, it is the cell wall of the bacteria. Okay, and then look at the structure number 3, pilus, or the plural is pili. Okay, This is the long hair. This is not flagellum, but it is pilus. So pilus usually present in some bacteria for conjugation, okay, or for transferring of the DNA between the bacterial cell. Okay, next number 4 is fimbriae. So this is structure fimbriae, almost similar to the pilus, but fimbriae is numerous and shorter than pilus. Numerous and shorter than pilus. And after this, we will look at the functions of the fimbriae. Okay, number 5, okay, nucleoid or nucleoid region. So this is the structure of nucleoid. Okay, nucleoid ni is a DNA. Actually, it's a circular DNA. Because of the bacteria has no nucleus, no true nucleus, so the DNA is suspended in the cytoplasm of the bacterial cell. Okay, number 6 is plasmid. So plasmid is something that is circular in shape here. So actually, plasmid is a small circular DNA. And number 7 is flagellum. So the singular is flagellum and the plural is flagella. So some bacteria has only one flagellum, or another there is some bacteria also that have more than one flagellum. Okay, so let's look at the structure, all the 7 structure in detail. So the first one is capsule. Capsule is a jelly like or sticky layer of polysaccharide. So actually capsule is made up of polysaccharide. It's a carbohydrate. Function is to protect the bacteria against dehydration or to protect or shield the bacteria from attacks by host immune system. Okay, next, the cell wall of the bacteria. Okay, cell wall of the bacteria is composed of peptidoglycan. Okay, it is not like plant cell that have cell wall made up of cellulose. It is differ. So the bacteria cell wall is made up of peptidoglycan. Okay, peptidoglycan is actually a combination between peptide comes from the word peptide is something about protein, glycan is something about carbohydrate. So it is actually combination between protein and carbohydrate. Okay, next, structure pilus, singular, or pili in plural. So like number 3 here, this is the pilus. So function pilus, as I said just now, it is assists in conjugation or for the transferring of the DNA between bacterial cells. Or pili also is used to communicate with other bacteria. Okay, it is used for bacteria to communicate with other bacteria by using pili. Okay, next number 4 is fimbria, the singular, or fimbriae, plural. Something that is almost similar like pili, but it is shorter and numerous hair. Lebih banyak. Alright, so functions of the bacteria is of the fimbriae is to help bacteria to adhere or to attach. Attach to where? To one another. Bacteria A and bacteria B, they are attached by fimbriae, or fimbriae is used to attach the bacteria to the surface of the cell. Okay, for attachment of the bacteria to the substrate or to the surface of cells. Number 5 is nucleoid region. Nucleoid region also can be known as DNA. So the purple color here is the DNA. So what are the characteristics of the DNA in prokaryotic cell? So the DNA is circular and not associated with histone protein. Okay, not like eukaryotic cell or like animal cell, like human. Our DNA is not circular. Our DNA is linear and our DNA is associated with histone protein. Okay, functions of the nucleoid region, of course, as a genetic material because it contains DNA. And number 6, plasmid. Okay, here the plasmid is a small circular DNA. Because of its size that is small, it carries only a few genes. only some bacteria have plasmid. Next is flagellum, singular or flagella, plural. Okay, for flagellum, it is different from the eukaryotic cell. In prokaryotic cell, the flagellum is made up of protein flagellin. Okay, differ from eukaryotic cell. And functions of the flagellum is for locomotion and for feeding of the bacteria. Okay, let's look at the structure of cell wall in prokaryotic cell. Okay, based on its cell wall, bacteria or prokaryotic cell can be classified into gram-positive bacteria or gram-negative bacteria. Okay, so examples of gram-positive bacteria is like lactobacillus species. So how is the cell wall for gram-positive bacteria? Okay, so this is the cell wall of gram-positive bacteria where it consists of thick peptidoglycan layer, thick here. Okay, compared to gram-negative bacteria, for example like E. coli and Salmonella species, so the cell wall of this bacteria, okay, this is the cell wall of the gram-negative bacteria, where it consists of two things. First is it has only thin peptidoglycan layer, and it also has outer membrane on its cell wall. So in terms of peptidoglycan, in gram-negative bacteria, it has thin peptidoglycan layer, but in gram-positive bacteria, it has thick peptidoglycan layer in its cell wall. Okay, other than that, in gram-negative bacteria, okay, at the outer membrane, there is a presence of this green color hair. Okay, this is what? This is lipopolysaccharide. So this lipopolysaccharide that will cause this bacteria, this gram-negative bacteria, become very toxic to its host. So compared to gram-positive bacteria, gram-negative bacteria is more virulent or more pathogenic because of its presence of lipopolysaccharide. Okay, so this is the example. E. coli and Salmonella species. Okay, now let's look at the comparison between prokaryotic cells and eukaryotic cells. So again, I remind, if we give comparison, we must have similarities and differences. Okay, there are three similarities between prokaryotic and eukaryotic cell. First is both cell is surrounded by plasma membrane. Okay, plasma membrane. Then both have DNA as genetic material, okay, of course. And the third one, both have ribosome. Okay, the only organelle that bacteria or that prokaryotic cells have is ribosome, because prokaryotic cell only have organelle that is not membranous organelle, or organelle without membrane. So the only one organelle that have no membrane is ribosome. Okay, so the differences, there are few aspects. So the first one, of course, in terms of its size, prokaryotic cell is smaller in size, while eukaryotic cell is larger in size. So this is the size, 1 to 5 micrometer for prokaryotic cell and 10 to 100 micrometer for eukaryotic cell. Okay, second aspect in terms of its nucleus. So for prokaryotic cell, it has no true nucleus, while eukaryotic cell has true nucleus. Okay, look at how it differentiates in terms of the same aspect. But if you want to differentiate in terms of the DNA, okay, let's look at the correct sentences. In prokaryotic cell, the DNA lies freely in the cytoplasm, while DNA in eukaryotic cell contains in the nucleus. So please take note if you want to differentiate in terms of the nucleus, you must have the same aspect for the eukaryotic cell, which is you give the comparison in terms of the nucleus also. But if you want to differentiate in terms of its DNA, so of course for the eukaryotic cell also you must use DNA as the aspect. Okay, but if you differentiate not in the same aspect, for example, prokaryotic cell you say about no true nucleus, while for eukaryotic cell you give DNA. So you will not get the full marks for this aspect. Okay, 0 mark actually. Okay, next aspect is about organelle. For prokaryotic cell has no membrane bounded organelle like I said just now. Okay, no membrane bounded organelle means dia tiada organelle yang bermembran. That's why prokaryotic cell only have ribosome, because ribosome does not have membrane. Okay, while for eukaryotic cell has membrane bounded organelle. Okay, in terms of the characteristic of the DNA, for eukaryotic cell, the DNA is circular DNA and DNA is not associated with histone protein. While for eukaryotic cell, the DNA is linear DNA and the DNA is associated with histone protein. So let's look at this aspect where if you want to differentiate both cells based on its DNA characteristic, you must give the complete sentences like this. Okay, you must explain about the shape, which is circular, and about the histone protein, which is for prokaryotic cell, it is not associated with histone protein. Okay, next. Okay, about the cell wall of both cells. Okay, cell wall of prokaryotic cell is made up of peptidoglycan, while cell wall of eukaryotic cell, okay, plant cell wall composed of cellulose, fungi cell wall composed of chitin, and animal cell lacks cell wall. Okay, why for this aspect you must explain about plant cell, fungi cell wall, and animal cell, because all of this cell is example under eukaryotic cell. So you must mention all type of cell. Okay, and lack in biological term, lack means no, absent. Okay, so animal cell does not have cell wall. Okay, next about the ribosome. For prokaryotic cell, even though it has ribosome, but it has a small size ribosome, about 7ts. The unit is 7ts, while for eukaryotic cell have large ribosome and the unit is 8ts. Okay, in terms of the flagella, both of course have flagella, but it has difference in terms of its arrangement and its composition. So the first about arrangement is flagella in prokaryotic cell lack 9 plus 2 microtubule arrangement. Okay, tiada susunan 9 plus 2 microtubule. And it is made up of protein flagellin. While for flagella in eukaryotic cell, it has 9 plus 2 microtubule arrangement. So this diagram is a cross section of the flagella. So where is the 9 plus 2? Okay, 9 plus 2 microtubule arrangement is like this. So this one is the first microtubule arrangement. 1, 2, 3, 4, 5, 6, 7, 8, 9. Plus 2 is microtubule at the center, ada 2. So this arrangement only present in flagella of eukaryotic cell. Okay, but in terms of composition, flagella of eukaryotic cell is made up of protein tubulin. Okay, if I am a student, actually I will prefer to memorize about the composition of the flagella. Okay, you just memorize protein flagellin for prokaryotic cell and protein tubulin for eukaryotic cell. Okay, next is a structure known as mesosome. Okay, only bacteria or prokaryotic cell will have mesosome. So where is the mesosome? Okay, let's zoom in this diagram. Okay. Okay, where is the structure of mesosome? So this is mesosome. Here. Okay, mesosome is a structure that comes from the inward folding of plasma membrane. So ni plasma membrane. So this is plasma membrane. When plasma membrane ni inward folding, maksudnya dia terlipat ke dalam, so it will become into a structure known as mesosome. So what is the functions of mesosome in prokaryotic cell? It is act as a site of cellular respiration. Site. Tapak for cellular respiration. Okay, so what is the site of cellular respiration for eukaryotic cell? If prokaryotic cell have mesosome, eukaryotic cell have mitochondria as a site of cellular respiration. Okay, so this is the aspect. Alright. Okay, next. How they divide. Prokaryotic cell divide by binary fission, while eukaryotic cell divide by mitosis and or meiosis. Some bacteria will have circular plasmid, while plasmid are absent in eukaryotic cell. Okay, please take note, if you want to answer structured or essay question during exam, please make sure for comparison, for differences question, you are not allowed to write down in table. Okay, for both structure and essay question, no table at all. But you must explain, you must write full sentences by using comparable word like while or but or anything that is similar to these words.